Jerry, you're on.
Okay, good afternoon. I'm Jerry Bartholomew. I'm a professor in microbiology and organizer of the OSU Art Sci group. Earlier this year, my co-host Dominique Bachelet organized a series of talks by scientists on the positive aspects of climate change in order to inspire artists for a show at the Corvallis Art Center that will be held in the fall entitled "What Will Nature Do?" So this term's series of talks is co-sponsored by the Art Sci Group, the Corvallis Art Center, and the Department of Biological and Ecological Engineering. And here we're focusing on scientists who are also artists, or artists who are also scientists, with the goal of exploring how their science might inform their art, how their art might inform their science, and how they integrate these. So today I have the pleasure of introducing a good friend, David Madison. David is a biodiversity explorer who fell in love with beetles when he was 15. And he has expressed that love through his research on the evolutionary biology of beetles, as well as through his art. For decades, he's wandered the branches of the evolutionary tree of life of beetles as he explored the evolution of their forms and genomes. He's currently the Harold E. and Leona M. Rice Professor in Systematic Entomology in the Department of Integrative Biology here at OSU and Director of the Oregon State Arthropod Collection. One of his passions is discovering and documenting the species of beetles that live on Earth, and he's done extensive fieldwork looking for beetles throughout North America, as well as Chile, Argentina, Ecuador, Australia, New Zealand, Germany, and Italy. But what brings him here today is that for one window of time in days gone by, and still occasionally, he was primarily a scientific illustrator of insects, and he continues to explore the art in his science in numerous ways. In addition to beetles and art, he's also the co-author of 2 widely used pieces of software in evolutionary biology, and he loves the puzzle-solving aspect of coding. So David, I'll hand it over to you now.
Thank you, Jerry. So, and thanks very much for inviting me. This is a talk unlike any that I've given before. And in putting it together, it's just really been a thoughtful time for me to ponder what to say. And one of the results was that I started to lose track of what the boundary was between my art and my science. And so let's see if I can explain why here. Okay, I'm going to share my screen. Okay, and we have this. Okay, we all good? Thumbs up? You can see the right?
No. We're seeing the presenter view.
There's the presenter view. Okay, that's not what we want. We want this one. Yes? Now
Yes,
we're good?
perfect.
Okay,
Perfect.
very good. Thank you. Okay, so I'm going to start with a story. And the story that I'm going to tell starts in South America, in particular in the mountains of southern South America around the Chile-Argentina boundary, in the land of volcanoes and monkey puzzle trees, and also beautiful, cold, clear mountain rivers. This one in particular, Arroyo Cañi in Argentina, is incredibly cold and clear. And if you were to go and look under one of the rocks on the shoreline, you would very likely find a little beetle. And that beetle, I don't know if anybody's looking at me, but that little beetle is that big. It's about 5 millimeters long. And let's imagine that we thought a little bit about that beetle and where it came from. And we're going to think in particular about where its DNA came from, where it got its genetic material. And so let's imagine that we look into one cell in the beetle's body and we saw the chromosomes in that cell. And let's say we zoomed in on one of the chromosomes and saw a stretch of DNA. And the particular DNA that we're going to focus on is 18S ribosomal DNA. And there's a little, there's the sequence. You can see the sequence in the A, G, T's, and C's at the bottom. So the question that I want to pose is, where did that come from? And what was its history? That particular stretch of DNA, that's a part of a molecule. It's made up of atoms. Where did it come from? Well, most immediately, where that stretch of atoms came from was the mother cell of the cell that whose chromosomes we're looking at. And that stretch of DNA arose from that mother cell through a process of DNA duplication. So the DNA that was in the mother cell was duplicated, and one of the copies was given to this cell that we're now looking at. Well, where did it, if we look back to that mother cell, where did it come from in the mother cell? Well, it came from the mother cell of that mother cell, and so forth. And we could trace through a series of DNA duplication events, simple molecular processes, that stretch of DNA, its copies, its ancestral copies, back to the egg that formed that beetle, to the zygote. And so what we're going to do now is go through a little journey back in time, imagining tracing the ancestors of the DNA, that stretch of DNA, even further back. And again, each of these ancestors is deeply connected one to another just by this simple molecular copying process. So just to make this a little bit simpler, we're going to make this diagrammatic. And so there's our beetle on the river shore. And let's imagine that we could trace the year before and look at its mother and its father. And we're going to, again, simplify this, and we're going to represent them as just dots so that the imagery is simpler. So this diagram right here shows our beetle in red and its two parents in yellow from the previous generation. And that blue line indicates that the stretch of DNA that we're thinking about in the egg of that red beetle was inherited directly from the egg that was produced by its mother. And we could track back within the body of the mother that stretch of DNA through the development of that mother to its zygote, and so forth. And so let's imagine this in the context of not just our one beetle now, but all the other beetles that live in that species in the year that we found that beetle, and all the other specimens that were present in the population the year before, the parental generation. And we could trace back through time to the grandparental generation. And we could imagine tracing back to see where that stretch of DNA came from in the grandparental generation. We could step back even further to the scale of decades and imagine tracing the history of that copy through its ancestors back through the population. Now, we can't actually do this. We don't have the scientific tools to do this perfectly accurately. We can infer some aspects of these histories, but let's just imagine that we're omniscient at the moment and could actually see this history in all of its detail. So let's imagine we step back not in terms of decades, but in terms of millions of years. And let's say we trace the history of that stretch of DNA back through time, millions of years, into the ancestry of that species. At one point, we can look over our shoulder and see a different species branching off from the ancestor and going forward in time to a related species. And so this is another species that lives in South America. We could imagine doing the exact same little game that we played with this one. Let's look in the body of that specimen, find a stretch of 18S ribosomal DNA, and we could trace its ancestry back of that individual copy back to the common ancestor of these two species. And the beetle in whose body that common ancestral molecule would lie, that stretch of DNA is the direct ancestor of the stretch of DNA that we sampled in this species and our original specimen that we looked at. Okay, we could go back even further, and let's imagine this is the whole tree of life. So here's bacteria, here's archaean, here's eukaryotes with animals up here. Here's our beetle. Let's imagine that we walked back in time, peered back in time, not just over the course of a few million years, but traced back in time that stretch of DNA through millions and millions of years. Well, we could do the same for a stretch of DNA in me. And if we pick a stretch of DNA, that same stretch of DNA, 18S ribosomal DNA, we could trace it back through the history of Homo sapiens, of primates, of mammals, of vertebrates, all the way back to a point in time in which the copy that was the ancestral copy of the copy in me is exactly the same piece of stretch of atoms as the ancestral copy of the beetle. Now that common ancestral copy of that one stretch of DNA would have existed sometime in the late Precambrian. And as I like to say, you know, when you give talks, you're always supposed to be perfectly honest. That's not actually a picture of the late Precambrian. That stretch of DNA would have, though, lived in, presumably in a body of some worm-like creature in the ocean. And I think it's one of the most amazing things to think about is that if you were to go back in time to that point and then go forward a few million years, what you would see is not one species of worm-like creature, but two species of worm-like creature that may differ in only some minor trivial way at the time. One might have some dots on it that the other one doesn't have. And yet those two different species of worm-like creature had such dramatically different fates that the one on this side eventually diversified and gave rise to all the insects, including this beetle. And the one on the other side eventually diversified into all the vertebrates, including me. And this idea that all of the creatures on Earth are deeply, deeply connected through these genetic connections, this simple process of DNA duplication, through this genetic network that's over 3.8 billion years old, that's connected via this tree of life, is to me one of the most profound insights that Homo sapiens has ever come to. And Charles Darwin has a beautiful quote about this tree of life. The affinities of all the beings in the same class have sometimes been represented by a great tree. As buds give rise by growth to fresh buds, and these, if vigorous, branch out and overtop on all sides many a feebler branch, so by generation I believe it has been with the great tree of life, which fills with its dead and broken branches the crust of the earth and covers the surface with its ever branching and beautiful ramifications. So I am in awe of the tree of life, and that inspired me to write a poem some time ago. I think that I shall never see a thing so awesome as the tree that links us all in paths of genes down into depths of time unseen, whose many branches spreading wide house wondrous creatures of the tide, ocean deep and mountain tall, darkened cave and waterfall. Among the branches we may find creatures there of every kind, from microbes small to redwood vast, from fungus slow to cheetah fast. As glaciers move, strikes asteroid, a branch may vanish in the void. At Permian's end and Tertiary's door, the tree was shaken to its core. The leaves that fall are trapped in time beneath cold sheets of sand and lime, but new leaves sprout as mountains rise, breathing life anew neath future skies. On one branch, the leaves burst forth, a jointed limb of firework growth, with inordinate fondness for splitting lines, armored beetles formed myriad kinds. Wandering there among the leaves, in awe of variance time conceived, we ponder the shape of branching fates and elusive origins of their traits. 3 billion years the tree has grown from replicators first seed sown to branches rich with progeny, the wonder of phylogeny. So the tree of life, the evolutionary tree of life, now has 1.8 million known leaves, i.e. individual species, and many, many millions more yet to be discovered. And the fact that the biological world has this tree-like history to it of genetic connections has profound implications and impact on the nature of the patterns that we see in the biological world. The patterns, for example, of traits between the species. And I realized at some point in my career that this is what I love. I love patterns. And the patterns created by the tree of life are my favorite. So, okay, so this is what I'm going to talk about in the talk. I've already talked about the evolutionary tree of life and its nature. I'm going to say a little bit about beetle diversity. I'm going to talk about my history with beetles and illustration. And then I'm going to give some examples of my life as an explorer of patterns. Patterns in nature, patterns with nature other than beetles, patterns within one beetle, patterns among specimens created by the boundaries between species, the patterns created by descent along the branches of the tree of life. And then I'll talk a little bit about some summary thoughts and end finally with a few notes about some of the artists around me that I think you might be interested in. Okay, beetle diversity. So beetles are amazing. They are just these wondrous little forms and colors. So actually, all the ones I'm showing you here belong to just one family of beetles. So there's 400,000 known species of beetles. The family I work on, the ground beetles, has about 40,000 species that are known. New ones being discovered all the time. And this family, the carabids, is the family I love the best. And I'm particularly fond of the genus Bembidion and its near relatives, and this is a Bembidion. And the more you look at beetle diversity, the more various forms and echoes of similar forms that you see. And the beauty in these creatures is not just in their overall form, it's as you start looking close up at them, you see all these amazing structures. So this is a head of one of the ground beetles. This is a different species. Here's a tiger beetle, a different tiger beetle. This is one of the snail eaters that lives around here. And all of these this diversity of form in insects has inspired artists and illustrators over the years. And in carabids, I just wanted to give a shout out to this species and one of the pieces of art that's been created for this one. This is Bembidion zephyrum, a species that's common on the Pacific coasts along here on the sandy beaches. And Jesse Smith, it's a species I work on, and Jesse Smith created a belt buckle that is based upon this species. Okay, so my history with beetles and illustrations. My history with beetles apparently starts very young. I don't remember this, but that is probably me pointing at a little beetle on the sand. The formative years were when we stayed at a cottage in the summer in Northern Ontario. This is me, I think. Well, I'm not certain which one's me. I've got an identical twin brother. That's my mom. And this is a book I had when I was 7 years old, and I was always interested in the natural world. And up at the cottage there, I did a huge amount of exploring with my brother. But the seminal moment was when we were 13 years old. Here we are as 13-year-olds. And we had moved across Canada and we're staying in a motel waiting for our house to be ready to move into. This is in Burlington, Ontario. And Wayne and I were playing on the shores of Lake Ontario on those rocks, and a pile of grass clippings floated by. And there on the grass clippings, Wayne looked up and noticed on the grass clippings a jumping spider, a Phidippus audax. And those eyes looked up at us and we fell in love. And to this day, jumping spiders have been a very important part of both of our lives. And they are among the most amazing creatures on Earth. Here's a pro tip. Google peacock spiders if you're not familiar with them, and watch YouTube videos on peacock spiders. So this is actually a species, this is Habernatus americanus. This is actually a species that lives in Oregon. It's actually on Mary's Peak. It lives in the Cascades. This specimen came from the Wallowas. There are only about I think they're about 4 millimeters long. So it's not as though they're giant, but you look up close and they're amazing. So there we were as 13 years old. We fell in love with jumping spiders. We got Spiders in Their Kin, the Golden Nature Guide. And one of the things that was amazing about this book was, as with I think all of the Golden Nature Guides at the time that were field guides, they had illustrations of the organisms, which was very inspiring. And we just wanted to find everything that was in that book. And we wanted to express our love for jumping spiders in any way we can. And one of the ways that we could was with art. And Wayne and I had always loved to draw. And we had various things to draw with, including colored pencils, and we had some inks. And we just started to draw. And this is the oldest scientific illustration I have in my portfolio, the oldest one I can find, and it's of the zebra spider, Salticus senecis, that I drew when I was 15. Now, Wayne has continued on with, these are some of his illustrations. The ones on the left is a page from his PhD publication. And Wayne is now a world expert on jumping spiders at the University of British Columbia. We have formed a great team over the years. I no longer work actively on jumping spiders. We've done field work together. One of the things that we've spent a lot of time together is this phylogenetic software, which we've developed. And as might not be too surprising, it's very graphically oriented. And here we are not too long ago, sitting in a nice place, coding together, trying to build parts of the software. But back to our youth, and the moment when we were actually not 13, but when we were 15, and I realized I needed to get something different. Jumping spiders were Wayne's. I needed to have my own thing because we couldn't both have jumping spiders, it became clear. And I fell in love with beetles, and in particular ground beetles. And I won't say much about that story. I'll allude to a little bit later on of that perhaps. But I also started picking up my tools to celebrate my love for the beetles. And this one right here is the oldest one that I've got that is a complete illustration of a beetle. This is a little ground beetle from the Southeast US and farther south that I did when I was 18. This is a tiger beetle. And so these are all the same method. They're basically layers. The bulk of the bottom layer is colored pencil with inks on top of that and with some modifications. So there can be colored pencils on top of the inks, watercolors on top of the inks, and so forth. So it's a mixed media approach. The wasp, paper wasp. When I was just after my undergrad days, I took some time off from biology per se and was a scientific illustrator and made a little bit of money through commissions, and that's what some of these are. And again, most of these insects are small, significantly like ½ an inch or less, or much more smaller. This is larger. This is a swallowtail butterfly and a thistle. This actually happens to be a species that lives around here, but that's a coincidence in my life. And then when I was doing my master's degree, I did an illustration of one of the beetles I was working on at the time. This is a Bembidion aliskensi. And this is probably what I view as my best illustration, best full color illustration. And it's one that fortunately actually saved pictures of the progress of the method. And so on the left is just an outline done with a crow quill pen. So that's just a fine. No, actually, this would have been done with a rapidograph. And then in the middle is a quick layer. I at the time used Pantone marker as a quick layer down to sort of color the paper. Very bad idea. Never do it again. I would do watercolor now rather than Pantone. The Pantone turns out damaged some of the other ingredients in the mixed media. Oh, wait. And then on the right is a layer of colored pencil, just pure colored pencil. On the left here is the colored pencil layers being refined. In the middle, the inks are started to be applied to the colored pencil layer. More inks applied on the right-hand one, including some general ink patches. And those ink patches on top of that colored pencil, it acts like scratchboard. And so you can actually scratch through. And there on the left here is it partly scratched to start forming the pattern of the ommatidia of the eyes. In the middle is one in which the head is mostly done, although as you can see, it doesn't have the hairs on the head yet, anywhere in the head. And then the last one is a close-up of the finalized drawing. The last full color one that I did, big full color one, was when my mentor, George Ball, turned 80 years old. And so in 2006, I drew an image of the beetle that he fell in love with when that led to his PhD in many years before. And so this is this is not a small beetle. This is a beetle that's maybe 1.5 inches long, and it's common, a beautiful thing that's common in the Southeast US, and the Midwest. So over the years, I've done a fair bit of pen and ink. These are actually straight scans of the plates, the way we used to do it when we submitted things for publication. So the direct plates themselves. So mostly stippling was the style that I used. That's a larva on the left. That's on the right, they're components of the male genitalia. And then this is an array of the male genitalia of one particular group of beetles. These are some coquille drawings. So this is a textured board that is basically like auto stippling, is one way to look at it. More recently, my illustrations have tended to shift towards digital, for better or for worse. And so here's a plate from a recent paper. And frankly, a lot of the stuff I do now is so deeply integrated with the scientific results that it has less of an organic sense than what my old ones used to have. Just as somewhat of an aside, I decided to try stained glass. And so here's the head of one of my beetles in stained glass. I've only done 2 stained glass pieces. So I have dreams of doing more scientific art stained glass things, but I haven't done it yet. And this isn't a scientific illustration, but that was my other piece. Okay. My life is an explorer of patterns. So as I said, I love patterns. And I clearly love patterns with repetition, especially ones that are in nature. And of course, the patterns aren't restricted to that scale. There are patterns when you look within the body of a beetle. So the little beetle on the left, which is about 3 millimeters long, if you looked where that white rectangle is and looked at it under a special type of compound microscope, you'd see what's on the right. And I could stare at these all day. So here's a different part of the beetle of that same beetle. And it's just lovely. It's absolutely gorgeous. Here's a beetle from South America, and here's the central body part of it. Here's that Bembidion zephyrum, the one that the belt buckle was made for. And if we magnify that part right there that's in the rectangle, that's what it looks like close up. And I get to spend my days just looking at this. Isn't this just so cool? This one right here, this group is common in Oregon and North America, actually common in Europe and Asia too. And this is what parts of the body look like close up in this one. And in fact, these close-ups, this interesting microsculpture, as it's called, inspired my daughter to create a puzzle. So she took pictures and made a jigsaw puzzle, which is actually a very interesting thing in terms of science communication, because to really be successful at it, you have to understand something about the nature of microsculpture and how the beetles vary in it. Okay, so this one is another beetle that I want to show you a couple close-ups of, and in particular, part of the leg. This is a beetle that lives on Mary's Peak. And this is a scanning electron microscope of what that basal part of the tarsus, the foot pads, as it were, of the front leg. And there again, you know, it's just this, it's like this, it's got this element of repetition, but with variation that is so appealing. And you could explore these bodies again also for as long as you want, and you'll just continue to find these amazing, beautiful patterns. These patterns aren't just on the outside of the body, they're on the inside. And I just wanted to explain something about these beetles. So they've got interesting male genitalia that are these tubes. So here's this tube, there's this tube on the outside into which is folded a membranous sac. So it would be attached to the body back here, and it's inside the body most of the time. So there's this, basically this clear, hard tube into which this internal sac, as it's called, is attached. And during mating, the sac gets pushed out down here. So here it is, pushed out. See this yellow bit here is that yellow bit there. And here's the sac pushed out. And because of the fact that this structure is involved in mating, it varies a lot between species. And actually, it's probably a mechanism whereby individual species can tell each other apart, and they know, and it ensures that specimens mate with something of their own species, which means that there's also actually tremendous variation in that body part and tremendous complexity of structure. So if you looked on the outside of that internal sac, this is what you see. And again, like just no end of interesting variation. So there's a lot of patterns within the body of one beetle. And again, it's just something that my mind is infused with as I do my work. And I think it plays a big role in terms of how I see these things. But there's also really interesting patterns that are created because of the fact that the biological world is organized into species. It's organized into specimens that interbreed, that are separate from other specimens with which they don't interbreed. So, and I'm going to tell a little story about this little beetle from Mary's Peak. So 3 days after I arrived in Oregon, I went up Mary's Peak and found this beetle. And I, over the next few years, realized that this little beetle, that this thing lived, there's my daughter, up on Alder Creek Falls. It lives on the edges of the waterfalls or on the edges of the nearby seeps that come down the rock face. And this waterfall Lineaepha, it's the genus Lineaepha, I had wondered, I thought maybe it was Lineaepha osculans, which is a very widespread common species. That was my first guess, was that these things I was finding on Mary's Peak were Lineaepha osculans. And Lineaepha osculans, and I thought maybe they are. I wasn't certain. Lineaepha osculans lives, those dots are where the known records of Lineaepha osculans. Oh, I see, I think I've got things out of order. And it turns out that the waterfall ones, the ones that looked similar, but I wasn't quite convinced they're the same, were known from a few localities too. And so I wanted to try to figure out, are they the same species or not? And so I looked at the microsculpture on the back. And it turns out that every specimen, every female of Lineaepha, the waterfall one, the microsculpture looked like this, like these little bricks. And every one of Lineaepha osculans, they didn't look quite like the same. They weren't quite as rectangular and brick-like. And it was absolutely consistent for every specimen. When I looked at the male genitalia, I saw the male genitalia of the waterfall species looked like this, and osculans looked like this. And I thought, well, there's some differences, but maybe that's just individual variation. I don't know. So I looked at more specimens. And I'll just let you use your pattern recognition engine that's built into your brain to see that there's things about this one, these two specimens of this thing, that are in common, like the shape of that structure versus the shape of this structure in these two osculans. the configuration here is much more similar between these two than between this one and this one. And you can see that there's this echo of consistent differences that you see in the microsculpture. You can also see it in the male genitalia. And then I looked at the DNA sequences, and the DNA sequences had that in parts of this one particular gene, 28S ribosomal DNA, there was a stretch that was the same in all the specimens I looked at of osculans, and the waterfall one. There's another stretch the same, but there was this big chunk that was different between every specimen I looked at of osculans and every specimen I looked at of Lion-EPA one. And this is much of my life is about this, is about finding a pattern and then seeing if that pattern, if I can see repetition of that pattern in other parts of the body or in the genome. And to me, I just get so excited when I hear a melody and then I hear it again being played by a different musical instrument. And the reason that that's the case is simply that that this waterfall Lion-EPA is a separate species. There's interbreeding within its lineage, but there's no genes that cross over to the related species Lion-EPA osculans, and because of that, they have evolved and diverged separately in these multiple ways, including the microsculpture, the male genitalia, and the DNA. Okay, I'm going to skip over these guys for time's sake. So I ended up deciding that that this waterfowl Lion-EPA was a new species, and gave it the name Lion-EPA tuluqua, which means waterfall in the Santiam language. Okay, so we could look at this not just between two species, but see if we can see these repeated melodies across multiple species, and we can. And so again, if you look at microsculpture, each species has specific microsculpture patterns. Each species has particular male genitalia patterns. Each species has particular DNA sequences. And this view into nature, in which what I look for is these patterns that are repeating itself across different character systems, is a pretty deep part of how I view the world, as this series of patterns. But there's another key part to all of this, and that is that this diversity that you see across species is not organized in a linear way. We can line the beetles up like this, or line their genitalia up like this, and we can think of the patterns of variation, but fundamentally, the patterns of variation are dictated by the shape of the tree of life. And so, because what happens in the tree of life is that if you've got a particular branch in the tree of life, and you've got at the ancestor of that branch, some characteristic arose along that ancestral lineage, it would have been passed to the descendants of that branch. And so everything within that whole branch would have that characteristic or some modification of it. And because of that, the traits of organisms are organized in a nested hierarchical pattern. And I'll show what I mean by that with these guys. So here are these coquille drawings of the species I worked on, for my master's degree. And if you actually map out just the overall patterns that you see there when you're looking at the backs of the beetles, that on this ancestral lineage, those little 4, what we call silver spots, arose on there, and they were passed along the branches to all the living species. And then at this point in this ancestor, these darker patterns on the back, which are absent from these two species, they arose along this lineage and were passed through evolutionary time to all the descendants here. And somewhere along this branch, the much more complicated patterns you see in these species arose, and they were passed upon to the descendants there. And that the pattern of descent with modification along the branches of this tree-like structure yields these nested hierarchical patterns of organismal traits. it turns out it's, again, not just in the bodies of the beetle, the external bodies, but it turns out all of these ones also have one less chromosome than all of these ones. And so another hierarchical pattern that arose was that along this lineage, the ancestral species lost a chromosome, and all of these ones then also have the missing chromosome. So we get these nested patterns. And this happens at all levels, including at the very biggest level. So let's go back to this beetle in South America, and let's think about it in the context of the tree of life as a whole. Oh, that's an interesting, the graphic disappeared. Oh, well. Okay, so let's think about all the organisms on Earth, and let's imagine them. We're going to have a more diagrammatic view of that tree of life. And so if we can imagine this right here is all of life, and we've got the organisms laid on top, and let's imagine that this is the branch for eukaryotes that arose from within all of life, and everything from this ancestral point forward in time through evolutionary time all inherited the common properties of eukaryotes, such as having a nucleus. And then sometime later, animals arose, and all of these millions of species have inherited the common properties of animals, including multicellularity. And then nested within that are the bilateria, the bilaterally symmetrical animals, which have many features in common, including being bilaterally symmetrical. And then within bilateria arose the insects, which have 6 legs. And then within that are the beetles, which have the hard wing cover at the back. And then within beetles are the ground beetles, which have these fancy defensive secretions and various other properties of them. And then within the ground beetles are the genus Bembidion, in which our South American friend Rose is. And at each, so basic, and then within there is actually one clade within Bembidion. So all of these this Bembidion has inherited all the traits of all of those deeper levels. And again, all of the organisms on Earth, the pattern of their diversity is this sort of nested hierarchy when you look at it. I wanted to just, I don't know if I have time for this, oh, we're running short of time, but I'll just do this quickly. I want to look at this particular South American one a little bit more closely. So there it is, nested within Bembidion. And it turns out that some initial DNA sequence data suggested that there was this, something happened, was this quartet, or this sextet of, things are moving too slowly here, right? The DNA sequence data showed that those 6 species were closely, what the hell's going on here? The DNA sequence data suggested that those 6 formed one branch on the tree of life, which to me was not a sign of the natural hierarchy, but was a sign, it wasn't this melody of nested structures, it was discordance. And the reason that it felt like discordance to me was that these 6 species looked so unlike one another. It included the smallest Bembidion in South America, which lives in desert salt flats, the biggest Bembidion in South America, and then this one that lives on cold, clear mountain rivers. And no other group of Bembidion anywhere in the world has that diversity. And so it was like music, but with this discordant element. It turns out, though, that as I look at the bodies of the beetle more and the side of the bodies, all 6 of those had a pit right there that was present in no other Bembidion on Earth. And let's just take a look at that part of the body. And there it is right there. And so there's where the pit is. We've removed a lot of the body, and that pit is inside these giant horns. And those giant horns are not present in any other Bembidion. And basically, those giant horns were one of these, that's the one trait that also echoes the nested pattern suggested by the DNA sequences. And so even in this case where there seemed to be discordance, there was also concordance in the structures of the organisms. So the bottom line is that life, the tree of life, has given us all of these nested patterns. And I spend most of my day thinking about those nested patterns. So I realized in writing this is that what my job is, I started wondering whether I was really a scientist or what I was a discoverer of found art. The concept of a found poem, where you can look in some prose and see something that you could extract and really it's poem, it's poetic. And I realized that much of what I do is playing with art, with found art, be it on the body of a beetle, being it in the repetitive patterns that are created by the boundaries between species and the gene flow within species, whether it's the patterns, the beautiful patterns created by the nesting of the tree of life. And so one of the consequences of this is that when I think about my science, I think about it graphically so deeply that when I write papers, what I do is the very first thing I do as I'm starting to think about turning some research into a product is do the illustration. So I don't touch the introduction. I do the methods and the acknowledgements, but I don't touch the introduction, the results, the discussion until I've got all the figures and tables figured out. And then with that sort of graphical vision of the paper, then I can start saying, okay, what am I going to say about this? There's lots more to be said about that, but I just wanted to end quickly with some things about some people around me that I'm excited about. There are 3 graphic novels that are being produced in my lab right now. One is by Lucas Morimoto on the story about Lineafa, that Lineafa Tuluqua, and he's basically recounting the history of research on this group of beetles, including the various people who've worked on it and some aspects of interesting stories to do with the discovery. So these are some of pages from what Lucas is working on. Arden Smith is working on a book that recounts the tale of a class that I taught in which students were embedded in biodiversity research and in which we discovered 2 new species. And so these are from the graphic novel that she's working on. And another one that I didn't have pictures of is by Olivia Boyd, a grad student in my lab. I've also had the pleasure of teaching scientific illustration multiple times and have seen all of the talent, some of which was unknown to the students at the time before they started. All of the talent in the students, both graduate and undergrad at OSU. And that's it. That's what I've got.
Wow, those were talented students you got there. Man,
Oh,
beautiful.
yeah. Oh, yeah. And that was actually just from 1 year.
Wow.
I hadn't, since I put this together a little too quickly, I hadn't actually managed to get the ones from other years in there yet.
Impressive.
And in the past, you've had actual student shows at the end of the term. How did you work that during COVID?
We didn't. We didn't.
That's too
No.
bad.
So I taught it in the winter quarter, so last quarter, and unfortunately, it was all remote. And so I've actually never physically seen
Oh,
the work. Well, some of them did digital art, and of course, I've physically seen them as well as anybody could see them. But the hands-on stuff, I haven't seen yet. So we're hoping to put a book together of over the years of the work from the class.
yeah, that'd be
Great.
really impressive. Cynthia says, please do encourage them to enter the What Will Nature Do show if any of them apply.
Oh,
That would be
right.
a cool thing.
Right. Good idea. Good idea.
Mhm. Yeah.
So
did
I
your
have a
parents?
question about your artwork. You had a very sophisticated techniques from a pretty early age.
Yeah.
Where did you learn to draw?
By picking up a pencil and a piece of paper and doing things.
And you learned how to put together different media.
I think that the media, so colored pencils was natural, right? Because we had lots of colored pencils, or pencil crayons as we called them at the time. Uh, that's what we had on hand. And the ink was added because of scales on the jumping spiders. And so that was easy to add this, you could add the sense of a scale by putting it on top. And then I started adding watercolor on top of the ink to give this, it gives us a little bit more of a luminescent sense to it, and allows the highlights to jump out a little bit more. And then I started adding the watercolors or Pantone markers underneath the whole thing to make it that you didn't have to have as many layers of colored pencils, that the layer didn't have to be as thick. And so it was all just, okay, what do we got? Let's just start doing stuff.
Wow.
So your parents never drew? You never saw anybody in your family drawing?
I don't think, I mean, maybe I can never remember seeing either of my parents draw a single thing in my whole life.
Wow.
They were into, mom loves gardening, and she loves the biological world. She's not scared of it. And so she would show us things, right? The biological world. And dad, he loved fishing, and he'd get us involved with bait and collecting bait for the fishing. So they encouraged us in the biological realm, but the art, there was nothing like that. There's nothing there. But one of the brilliant things, I think, one of the absolute brilliant things is that we had each other. And I think that this is critical, because there are so many cultural messages to tell people that they can't do things, that you're not going to be good at, people aren't good, you're not, it's like a thing to not be good at art. It's a thing to not be good at math, right? And we didn't get those messages because we encouraged each other, right? And I think that played a big role in our just willingness just to play, right? And just to, like, it was all play. It reminds me of that song. There's a song, I forget who wrote it, but Eric Bibb has the most well-known. It's called The Cape. And it has one line in it that he did not know he couldn't fly, and so he did. And so I think that's, I think it's natural. You give kids just a bunch of tools, and they'll come, as long as you don't tell them they're not able to do something, they'll just come up with all sorts of stuff.
Yeah. So how many students do you have in your class on average, every term?
So let's see, on average, so what's tended to happen, so very quickly, I think I've taught it 5 times, I think, if I remember correctly. And the first couple of times it was reluctantly, it wasn't part of the plan. But the grad students in my department sort of grabbed me and said, please, please, please. And then it became a regular course. And the moment it became a regular course, I had to put constraints on it. Like it filled, there were constraints by the size of the class that it was taught in. And so I capped it at 22 or 23, and it would fill up. This last time, so in the winter, I put a cap on it of 25, just so that I could manage it a little bit more easily. And I had, I think, 58 applicants.
Wow.
So, I mean, one of the things that's absolutely clear is that the desire among students to do things that are at the art science boundary, either science students who want to have contact with art that is something that's comfortable and familiar to them, that has a biological component, or art students who want contact with science. There are just, I bet there would be hundreds and hundreds out there, right? That crave this.
Cool. Very cool. So you learn pretty much by playing with your brothers. So how do you put a class together to explain how to draw properly?
Well, so one of the things that I do is that I make it very clear to the class really probably should be called things that David knows about scientific illustration rather than a general scientific illustration class, because I've never been formally trained, right? And so there's lots of things I don't know about scientific illustration, but I've had enough contact with illustrators to, you know, have a fairly good idea. So the class is much more about it's much less about a lot of different techniques and more principles, right? And so just as example, one of the core principles I think is that that many times it's better, the best scientific illustration for a task at hand is to not draw what you see. Because if you draw, in other words, what you're trying to, the point of a scientific illustration is to, you have to define it very clearly. It's to convey some information, and you have to define what that information is, to an audience. You have to define what that audience is, right? And that audience is going to be a person, likely, presumably, right? And you have to think about human cognition when you do that. And so there's little tricks of how you draw things, including with pen and ink, that if you draw it the way you might think to draw it, it doesn't actually look, it doesn't go into our brains the best way, right? It's not that if you do things different, like a classic is, I don't know if I've got any here that are like that. Well, I've got some, well, it doesn't really matter. We don't need to, we're not doing a lesson here. Anyways, there's just, the point is that there's principles like that we go through. And that's kind of the main point, but it's also just to have for the students to explore on their own, right? And one of the things that's happened every year is that there's a talented group of students, and so they teach each other, right? Is that I very much encourage, like, we find out where people's skill sets are. There's some people who've never done anything like this before, which is totally fine, right? But the class doesn't expect you to start as an illustrator. And so, but there's inevitably people who are very skilled, who have different skills than I have. And so it's really a community effort.
Fabulous.
That's great. I wish I had
an option. Yeah.
Well, sorry, what was that?
Go ahead, Dominique.
No, Jerry, go for it.
Oh, I was just saying, I remember that golden book on insects, and I wish I had known you and your brother when you were kids, when we were all kids, because I wanted to write an insect book, and you guys could have been my illustrators.
You wanted to write an insect book.
I did. Cool. I don't know where that came from, but I clearly remember.
Jerry,
yeah,
it's
third
never too
grade,
late.
Maybe.
Jerry, it's never too late.
Now I'll write a Mixozoan book, and you would write
right.
Mixozoan patterns. So
right.
Next week.
So, you know, one of the things in thinking about this, I've thought about this whole pattern issue a long time, and realized that I'm atypical in one sense, in that a lot of scientists are more process people, and I tend to definitely be more of a pattern person. And I'm somebody who's less about hypothesis testing and more discovering the patterns, right? And to the extent that I deal with hypotheses, I create hypotheses rather than test them. And so, but as I alluded to at the start, the thing that I thought was so interesting as I was doing this was just thinking, my God, it's all art in a way, right? Is that, am I really an artist or am I a scientist?
Cool. You would be a good modeler, though. You know, testing hypotheses. You create your own hypotheses and put the patterns together. That's why you like writing code.
Yeah.
Very cool. Okay. If anybody has questions, you can unmute yourself and ask your questions.
I have a comment. I was at Oregon State starting about probably close to 60 years ago. It was a long time ago. And they have a long history of not only entomology, but scientific illustration. I started out working at the entomology department when I was 16.
Wow.
And been in 4-H entomology. And so I was always into anything that crawled around, and especially if it had anything to do with water. And so anyway, I was going to get a master's and I was going to get a bachelor's, and then eventually it turned into a master's in entomology. with a minor in art, but the art, I was working at the entomology department. The artwork took too much time, so I dropped the minor after I was about half done. But they did have scientific illustration classes, and I took, I think, a couple of terms of it. I don't remember who taught it, but I had a really good teacher in Corvallis. I grew up in Corvallis in junior high and was really into art, great classes, and that's where probably it all started.
Wow.
So there's a long history in Corvallis with, like I say, entomology and in particular the scientific illustration. But we didn't know anything about genetics other than I think we knew there was DNA, and we'd heard of Watson and Crick, and that was about it. And so what you can do now is just amazing.
Yeah. So do you know Bonnie Hall?
Yes.
Or did you know Bonnie Hall?
Yes.
She wasn't the one that you learned from, was it? Was she?
No, she was working for the entomology department.
Yeah.
And then her husband was in fisheries.
Yeah.
Was a professor in fisheries. And then she did a lot of illustration for the entomology department.
Yeah. Yeah.
And
I actually have those. Her husband.
She did a few things for my master's, and then she did probably about a third, and I did about probably 2/3 of the illustrations in my master's.
Yeah.
But yeah, she was working all the time doing scientific illustration.
Yeah. I didn't realize there was a scientific illustration class way back when. That's very cool.
I'll have to go back and see if I don't know who taught it. I can't remember.
I'd be very interested if you can track that down.
Well, I probably have transcripts in this paper blizzard in my office somewhere. I'm still doing art all the time, but I'm retired from a long career with the Forest Service. So,
Still in
I had
Corvallis?
some great schooling to get to that point.
Still in Corvallis?
No, I'm up in Puget Sound, just on the start of the Olympic Peninsula.
Ah. So we've got two Washingtonians tonight. We have Olympia and we have you.
My parents were in Corvallis until a couple years before they died. And so that was just a few years ago. And so anyway, we were always back in Corvallis helping them, usually for quite a while. We were there for about a week out of every month because they couldn't drive anymore. So I spent a lot of time in Corvallis. Holidays were always in Corvallis.
Mm-hmm.
And if I had my first choice, I'd probably be living there right now.
Oh, Puget Sound is beautiful, though.
It's nice. It's nice. Well, I worked on aquatic insects on caddisflies because they had described the adults, but no one had a clue what the immature stages look like. And I would love to talk to you, David, about Mary's Peak, because there's some very odd things on Mary's Peak with caddisflies.
Mm-hmm. There's a lot of strange things on Mary's Peak. That rock face with the seeps, that's got a huge number of very interesting things on it.
We always figured it was because that was the highest point in the Coast Range. So some things that were able to still survive there couldn't deal with the warmer temperatures. Then I don't know if some of the things that were there when I studied them 50 years ago are gone now. Who knows?
Yeah.
Cool. Very cool. Olivia, Gail, Jeff, any question? Jackie?
Well, thanks for coming, everybody.
Very interesting.
Yes, thank you.
Thanks.
Yeah, thank you. This is a lot of fun.
Yes,
Okay.
it was great, David. Fun too.
Good.
The other story.
Glad you enjoyed it.
Yeah.
And that's 50 years.
Yeah. And thank you for participating to this series. Much appreciated. Your willingness to do this.
Yeah. Well, you're welcome. It was fun. It was a
Great.
good thing to do. Okay. Good.
All right.
Okay.
Goodbye.
Bye.
Thank you
Bye.
so much.